Hey, it's been a while since I've done a video. First off, let me just apologize for the audio quality. I'm using my cell phone microphone instead of the one I just broke, which was my only working microphone. So it might be a little hard to pick up my voice, so I'm a little closer to the camera than I would be normally. Okay, so what is the purpose of this video? This is just going to be a short science demo video where I'm going to talk a little bit, do a quick experiment, and then hopefully in the next one I'll move on to something a little bit bigger. So in this video, I have a sample of ocean water which I collected from the Atlantic Ocean a couple days ago. I was drunk. I don't know why I collected the sample in the first place. I'm sure I had my reasons. I was already at the ocean and I had a bottle of water with me, and I was like, you know what? It would be nice if I had a sample to take home with me. It's normal. Hashtag normal. Okay, so I have my sample. I have a control sample here of purified distilled water for comparison as well. The two things that I want to discuss are specifically in regards to pH and salinity. The only experiment I'm doing is I'm going to be measuring the pH of both waters. My initial prediction is that the oceanic water is going to be basic, the purified distilled water is going to be neutral, pH of 7. I cannot measure the salinity of the water because I don't have the tools available for that. I could theoretically measure the density, but my scale is not that accurate, so I don't care that much. Okay, so, what is pH? Well, if you ever had a very basic explanation of any chemistry concept in high school or even middle school, they'll actually talk about pH. pH is nothing more than the measurement of how acidic or basic something is in solution. It works by measuring the concentration of hydrogen ions, H plus ions, or hydroxyl ions, OH minus. The tool that I'm going to be using is nothing more than something called universal pH paper. I've used it in a couple previous videos. The way it works is we simply put a little bit of the solution onto these strips of yellow paper, and we compare it against a color chart over here, and this will tell us roughly what the pH is. Very simple. I don't know the actual chemical reaction occurring in pH paper. Might be some sort of acid-base reaction, I have no idea. So we're going to be measuring the pH, and I will be discussing salinity after that. So let's go ahead and test the pH. Remember what I said? The ocean water should be slightly basic. I don't know how basic. The distilled purified water should be neutral. So let's take a quick gander at that and see what happens. I'm going to be using one straw to contaminate both of my samples, but that really shouldn't matter in this case. So, alright, this is, in fact, the ocean water. We'll now place a little drop. Oh, that's not nearly as much as I need. There we go. Okay, after placing a drop on the pH paper, there is no color change. Oh no, there is. It is a little green. Okay, so it's a little hard to pick up on the camera, but based off the pH paper in the strip, sorry, wrong way, and the color chart, we can actually see that the pH of the ocean water, while not 7, is actually between 5 and 7. This lends credence to my theory that the ocean water should be slightly basic. Okay, It's just a rough estimate. Let's go ahead and measure the pH of the other one. The other one should be much closer to the color for 7, which is a light vomit green. So I can just contaminate it. Okay. The color change, while difficult to pick up on the camera, is much more plainly visible to me. And we can see, or at least I can see, that it is significantly closer to 7, indicating that the acidity of the purified distilled water is actually higher than that of the ocean water. It is very, very hard to pick up that tint of green on the camera, unfortunately, especially since this is a yellow background. So you're just going to have to take my word for it. You can do your own experiments and find out yourself. The difference is minute, but it is there. Okay, so we tested the pH of both water samples, and the oceanic water was determined to be slightly more basic, maybe by 1 or 2 pH at most. This makes sense if we keep up with the news and learn that most ocean water should be relatively basic. Okay, so let's talk a little bit about oceanic acidity and the negative impact it has on the environment. If you ever kept up with the news in the last few years or so, maybe 5 or 6, you'd probably hear that the oceans are becoming more acidic. We just took a sample, and the sample that we took showed that it was clearly basic. This is true, and so is the previous statement that the oceans are becoming more acidic. It's only by a very small amount that they are becoming more acidic, and this is actually incredibly detrimental to most marine life. The pH balance in the oceans is something that microbes, as well as the major organisms in the oceans, have a delicate balance and require to be maintained at a certain level in order to live. If the pH changes too rapidly, as does things like the ocean temperature, things will start to die very, very quickly. Now, there is, of course, other factors at play. There are other factors at play. A brief segue I'm going to go into is another concept called salinity. We just talked about pH. pH is the measurement of how acidic or basic something is. Well, salinity is the measure of how much salt, sodium chloride specifically, a solution has. The oceans are incredibly salty. If we take a look at a chart, we can actually see that the brine levels in the ocean are pretty much the saltiest liquids on Earth, with the exception of, like, the Dead Sea, by a factor of a significant number. I can't even read that. So, here's the kicker. If we take a look at a cell, in order for that cell to survive in its surrounding environment, the conditions outside of the cell have to maintain equilibrium to make it easier on the cell. If conditions are salty outside the cell, that means that water will be pulled away through osmosis from inside to outside the cell, potentially killing it. The reverse is true as well. If the conditions inside of a cell are too salty and they're not on the outside, it will actually travel out the cell. So, if the saline levels of the ocean are that high, how do organisms survive? Well, evolution. Most organisms that survived in the ocean evolved to do so. In fact, one primary theory of abiogenesis is that life originated in the oceans. Although most land creatures would not be able to survive in aquatic environments as hazardous and dangerous to them as the ocean. They can survive in lakes and streams and ponds, but there are very, very few creatures that can actually... Turtles are an example, that would be able to survive, on shore and in sea. So this leads me to a theory that I have. Even though saline levels are incredibly difficult for life to thrive in, it seems that temperature and pH levels are actually more detrimental to marine life than saline levels. This is interesting. So what I think I'm going to do is, before I even get to that, in this little sample that I collected here, maybe 300 milliliters, they're going to die off eventually, but there is a ton of microbes and living organisms and cells inside of here. They're alive still. I only took the sample a couple days ago. They're not going to die yet. It's a closed environment. When they run out of nutrition and food, then yes, this will become nothing more than a sample of salt water. But right now, this is a thriving ecosystem. It doesn't look like it, but it is. So this brings me to my theory. I'm going to take some bacterial culture samples that I think I'll order, and I'm going to see how they do in various conditions of different water types. I will see how they thrive in saline conditions, in pH, and with adjusted temperature controls. My prediction is that the bacteria, because they're relatively simple organisms, will thrive in any condition where the saline is irrelevant. In other words, the saline levels don't matter as long as a constant pH and temperature is maintained. Any adjustments made to those variables will result either in cell death or the culture not growing as fast. That is my theory. Anyway, the next video hopefully should have more content and more experiments. Thanks for watching.